Now you're aware of that what are proteins, fats and carbohydrates and what are the sources for this. So now you have to perform test for the detection. So first of all, the common procedure is just the preparation of paste. You have to crush the sample to be tested and add water to the sample, then filter the solution. For every procedure you have to use this common procedure. So let's perform our procedure. So first of all, let's do the test for the presence of starch. So the procedure is you have to take a potato and cut it into 4 or 5 small pieces. Grind the pieces well. Then you have to take 2 test tubes A and B. Test tube A, add some water in both the test tubes, in test tube A and test tube B as well. And then you have to add some sodium hydroxide, or you can change the material and just wait. Like you can add, first of all you have to add the pieces of the cutted water or the paste you have made already in the common procedure. Add this potatoes paste and shake it well. So the next step is now add a few drops of iodine solution in both the test tube, and you have to observe any change in color. So you have to wait for the change in color. So here you can see the test tube A is changed blue black color into blue black color, and the test tube B is not even changed. The color of contents in test tube A changes to blue black. This shows that there is some presence of starch. So here you can diagnose that here is a starch present, or you can say the carbs. You can say carbs are the carbohydrates. So B is, so there are the blue black color indicates the presence of starch in the food. This is the result of our experiment. So the next experiment is a test for the presence of proteins. So let's perform our procedure. So take a test tube and put a small potato of egg white in it. So you have to put the egg white carefully in this test tube. So put carefully it into the test tube, just adding it. Now add a few drops of concentrated nitric acid in the test tube, or you can add sodium hydroxide instead of nitric acid. Now heat the test tube on the Bunsen burner. So now you have to remove the test tube from the flame and drain the solids content of the test tube into a petri dish. You can see now you have to put a few drops of ammonia hydroxide, or you can put copper sulfate drops in it. Observe any color change. So you can see the color of the egg white changes to yellow on addition of nitric acid, and change and orange on adding ammonium hydroxide to it. So if you are adding copper sulfate or sodium hydroxide, then it will change into purple to violet. So the result is the change in color from white to yellow, and then orange shows the presence of protein in the egg white. So here you are confirmed that protein is present. So next test is for the presence of fats. So the procedure is take a white paper, take a few peanuts and press them between the folds of the white paper. Here I'm showing that how you can press them. So you have to press them gently with the white paper, and then you have to observe. Now let the white paper dry for a while. So you have to wait, and you can observe that the white paper becomes translucent and an oily patch is pressing on it. So this oily patch results us, and they confirm that the oily patch on the paper shows that peanuts are present there. So here I have listed some common procedures for the test for proteins, and like you have to paste them, you have to add sodium hydroxide, you have to add copper sulfate, and then you can confirm. Then I have listed the test for fat, like you have to just rub the paste or the peanuts against paper, and you have to confirm. Then the test for starch, here you can take a screenshot here, and then you can test it on your own. So here you just have to add iodine solution, and you have to observe the color change. If the color is changing, then the starch, or you can say the carbs are present. So you can say carb is a second name for the carbohydrate. So if you are listening that there are carbs, so they are talking about carbohydrate.